So a much smaller group, and this is a principal component, the first principal component, basically showing that all cell counts tend to go up or down together. There's a lot of correlation between those measurements, but you can quite successfully partition the total number of cases. The 200 cases of major depression can be more or less cleanly separated into a group here with relatively high cell counts and another group with lower cell counts. And the group with the higher cell counts has higher scores on the Hamilton Rating Scale for Depression, which is a physician-rated depression score, and higher scores on the Beck's Depression Inventory, which is a self-rated scale of depressive symptoms. So higher immune cell counts as a marker of innate immune activation associated with increased depression scores within a cohort of patients, all of whom have a diagnosis of major depressive disorder. So I think what this result speaks to, which wasn't so obvious from the case control transcriptional results that I showed on the previous slide, is that within MDD, within that big clinical syndrome, we should be thinking about whether there's a subgroup of patients with a particular evidence for an inflammatory pathogenesis. Some other work that has been going on, this is from Athena Araldus, who was a PhD student in my lab until last year. This is looking at imaging markers in relation to inflammatory markers in patients with depression. And this is a resting state fMRI scan showing a system of nodes in different parts of the brain which are differentially connected in patients that have high levels of CRP compared to healthy controls. You can look at those data in a slightly different way. So this network, it turns out, is sort of functionally specialized for interoception. And if you look at the connectivity in that network overall, in relation to concentration of CRP in the blood, you can see higher levels of CRP, greater inflammation, reduced functional connectivity in this interoceptive network. And this is a plot showing actually the same kind of features, so changes in connectivity, but here in a sort of topological representation rather than an anatomical representation, and highlighting the difference between those two subgroups I showed on the previous slide, the patients with high cell counts, high immune cell counts compared to low cell counts. And you can see in the patients that have more immune cells in circulation, there's by eye evidence for greater decreases shown by these blue lines in connectivity between principally the nodes in the default mode network and the ventral attention network. And it's not just functional imaging that shows changes in brain states related to inflammation. You can also look at structural data. And when you think about structural MRI, we tend to think about metrics like volume, surface area, thickness. These are all macrostructural geometric measurements that you make on fairly chunky bits of the brain, subtending multiple voxels in an image. But there is also, as some people will know, a kind of growing interest in so-called microstructural MRI, which is using the versatility of MRI to measure something about tissue composition at an individual voxel. And one such microstructural MRI marker, which actually has been around for a long time, is proton density. It's basically just a measure of the amount of water in a voxel. And if you look at the relationship between proton density and C-reactive protein again, in patients with depression, you can see that there is an area, particularly in the posterior cingulate, where higher proton density, higher water concentration in these voxels is associated with increased inflammation. And intuitively, that doesn't really surprise me. If you think about inflammation in other tissues, is generally associated with extracellular passage of water, accumulation of extracellular fluid or edema, and the idea that in people with higher levels of peripheral inflammation you get a little bit of extracellular edema in the brain is consistent with these data. It's consistent with how proton density signals have been interpreted in other brain disorders. And this area of posterior cingulate cortex where we're seeing this microstructural change is obviously anatomically aligned with this kind of system. This is a largely default mode network of reduced connectivity in functional MRI data in patients with depression compared to control. So this is a kind of like a depression-related functional abnormality that involves the same brain region where we're seeing a microstructural change in relation to inflammation. So these are all kind of interesting correlational results. And if you think, well, that's provocative, could it be that the inflammation is causal to the depression? If that was true, then anti-inflammatory drugs should have antidepressant efficacy. And there is already some circumstantial evidence for this, and I say it's circumstantial because although it's quite robust and highly replicated and based on large samples, these are all data that were acquired and collected for a completely different reason. These are the same data that I showed you already. The primary purpose of these randomized clinical trials was to test anti-inflammatory drugs for efficacy in physical disorders like rheumatoid arthritis, ankylosing spondylitis, etc. But in each of these trials, the protocol included secondary endpoints like the SF-36, which you may have heard of as a general instrument that you can use to measure. It's often used in industry trials as a sort of general measure of patient well-being as a sort of secondary endpoint, but it does include two very simple questions about low mood and capacity for pleasure. And if you analyze those data using that SF-36 secondary endpoint as if it was the primary endpoint, as if that was the purpose of the trial to test whether there was a placebo-controlled treatment effect on low mood and anhedonia, it turns out that there's obviously quite a lot of variability between the different trials, but on average, over all of these studies, as I think about 12 studies and 7 different mechanisms of action, the average treatment effect is about 0.3, which is roughly the same as the average treatment effect of SSRIs or more conventional antidepressants. And for some mechanisms of action, it seems to be higher. The usual concern people have about these data relates to the fact that they are post hoc, unplanned analyses of trial data that were collected for a completely different reason, and people often think, well, maybe you're seeing an improvement in mood with an anti-inflammatory drug that you're offering to patients with rheumatoid because their physical condition is improving as a result of the treatment, and there's a sort of psychological reaction to that physical improvement, but it's all happening in this sort of psychological space and doesn't have anything to do with the drug mechanism of action. And that's a difficult counter-argument to completely address post hoc, but certainly we tried and failed to account for these effects in terms of physical health improvements. If you do the same analysis, for example, using only those participants that did not satisfy criteria for physical health improvement, you still see improvements in low mood and anhedonia. So these sort of considerations have stimulated quite a lot of interest in the idea that we might more deliberately and purposefully seek to treat depression using anti-inflammatory or more generally immune-modulating drugs. And if you're interested, this review that I published with colleagues from Janssen last year is quite a good sort of comprehensive analysis of the state of the field. And one of the things that really jumped out at it from my perspective was the number of plausible targets. I mean, there are many, many different places where existing drugs or new drugs can modify immune-related pathways for which there is some evidence of involvement in depression. And this is one particular target that we have been interested in the past. It's called P2X7. It's a purinergic receptor. So its endogenous ligand is ATP, which you'll be familiar with as a metabolic molecule, but it also has a role in the nervous system as a signaling molecule, and it's stress-related. So when you expose an animal or a human to high levels of biological or psychological stress, you get increased levels of extracellular ATP, which open this ion channel. Calcium comes into the cell, and if that cell is a microglial cell, part of the brain's own innate immune system, it activates the inflammasome, which ultimately releases cytokines like IL-1 beta into the immediate environment. So this is very, at a slightly simplistic level, this is a very intriguing target, because if you could block it, and there are drugs that can block this channel, in a way you would uncouple stress from immune response to stress. And that might be of interest in depression. So there are these data, correlational data. There are these meta-analysis results from previous clinical trials, all of which are kind of encouraging us to think that maybe there's a more substantial association between depression and inflammation, and indeed a prospect of novel class of treatment of depression. But I want to dig deeper into the question of causality, because you really, if you think about it from a drug development point of view, you really don't want to be investing a lot of time and a lot of money in a new drug to target a mechanism which is not on the causal pathway to the disorder you're trying to treat. And it is, something's gone wrong with this slide, so you'll just have to kind of ignore the bottom bit for the moment. But, everything I've said so far is pretty broad brush in terms of what's happening with the immune system, and pretty broad brush in terms of what's happening psychologically. And I haven't shown you anything which is more than correlational. And what we really need to do is kind of understand a bit more. Is there evidence that the innate immune system particularly causes depressive symptoms? And are any of these depressive symptoms or depressive-like symptoms particularly sensitive to innate immune effects? Anhedonia, fatigue. There's a sort of atypical depression syndrome that Brenda Pennings and others have described, of overeating, oversleeping, overweight, brain fog. Sickness behavior is the kind of animal model of depression, or one of them. If you give an animal LPS, or you give an animal cytokines, you immediately change its behavior, and it behaves as if it was depressed. And of course, it's also worth bearing in mind that all of these depressive symptoms are not unique to major depressive disorder. They can cut across various different diagnostic categories. So we need to think, I think, a little bit more about what we mean by depression in relation to the innate immune system. And we need to think, suppose there is a causal relationship between innate immunity and depressive symptoms. How is that mediated in terms of the brain? And what are the upstream factors that cause the increase in innate immunity in the first place? And there are many factors that can increase inflammatory state, obviously comorbid infectious or inflammatory disease, long COVID, obesity, social stress, and early life adversity. I'm particularly interested in the early life adversity story, and I'll say a bit more about that in a minute. But if you just follow the thread that in early life you could be exposed to an infection, or you could be exposed to a social shock, which perturbs your immune system and ultimately puts you at increased risk of depressive symptoms. You've got to interpolate a memory in the system somehow. The innate immune system is not where the memory of the immune system is. The memory is in the adaptive immune system. And so you get to this rather more complicated model where you've got inflammatory risks impacting first on adaptive immune cells, T cells, B cells, that control the state of the innate immune system and have effects on brain systems that ultimately lead to depressive symptoms. So it starts quite simple, and as you think it through, it becomes more complex, but still tractable. Now, why did I hit on early life adversity? Partly because it's such an important and well-documented, well-replicated and robust risk factor for mental illness generally and actually for physical health over the life cycle. And these data, which is from the Dunedin longitudinal study, are quite thought-provoking. So this is a cohort that was followed up prospectively from, I think, the middle of the first decade for at least 20 years subsequently. And the importance of the prospective nature of this design is it means that exposure to adversity in childhood was documented contemporaneously, whereas very often people try and link childhood adversity to adult states using a retrospective measure of adversity exposure, which is full of recall biases and various other confounds. So this is an exemplary study because the adversity exposure was well documented at the time that it happened, and then the individuals were followed up for long enough to see what the consequences of that were. And you can see that if you had more than two adverse childhood experiences as an adult, you were at increased risk of major depression, increased risk of physical disease, increased metabolic risk, and increased CRP. So more inflammation in longer, in adult life following childhood exposure to adversity. Now, what could be the molecular mechanism, or what's the mechanism of this long-term effect? And this is where it gets into epigenetics, which, as Sarah Jane said, I am absolutely not an expert in. But I am very intrigued, because if you look in the animal literature, and Elizabeth Binder in Munich, for example, as a leader in this space, there's two things jump out. One is there are a lot of different ways in which gene transcription can be controlled, moderated, modulated by various different epigenetic mechanisms. So epigenetics is not one thing, it's a family of mechanisms that control gene expression. And secondly, in animal models, there's abundant evidence that early life stress, or trauma in particular, can cause epigenetic modification. And Elizabeth Binder and colleagues in particular have worked a lot on epigenetic modification of the glucocorticoid receptor gene and another gene called FKBP5, which is a chaperone for the glucocorticoid receptor, and shown in a number of elegant studies that animals that have been experimentally traumatized show modification of these genes, which in turn affects, amongst other things, their neuroendocrine status and their inflammatory status and their behavior. Now, although the animal literature is very strong, the potential relevance of epigenetics for mental health has so far not really translated much into humans. And I think one reason for that may be that usually with an epigenetic study, you want to see how the genes have been modified in the tissue that's generating the symptom, if you're thinking about it from a clinical point of view. So ideally, or at least by first intent, if you're thinking about epigenetic mechanisms of depression, you might want to look at DNA methylation or histone modifications of neuronal genomes. And, we're obviously not going to get access to neuronal tissue in people with depression at any scale. There are some post-mortem studies, but, you're talking samples in the order of hundreds, rather few large collections. They're all post-mortem. They're mostly post-suicide. So it's a very complicated business trying to work out epigenetic mechanisms from neuronal tissue in psychiatry. What has been happening more recently is people have just been taking blood and looking at epigenetic modification of blood cells. And it turns out that you can quite often see changes in epigenetic status of immune cells in patients with depression or people that have been exposed to stress. So it's measurable, but then the question arises, well, okay, is it relevant? You know, how is it actually going to be helpful for us to think about epigenetic states in the tissues that we can measure? Are we just looking under the sort of spotlight, as it were, to find what we're looking for because that's the only place we can look? And that gets me to this study, which again is work from Mary Ellen Lionel that she did published last year. And this is an interesting example of open science. So nothing here was collected in Cambridge or, this is a study that could have been done anywhere in the world. But it has an interesting question motivating it, which is if you look at genomes from multiple different tissues in the body and you look at their epigenetic status, is there any evidence in any of those tissue types that the regions of the genome which are epigenetically activated are disproportionately co-located with known genetic variants conferring risk for psychiatric disorder? So you take a lot of GWAS studies and you collect the list of risk genes that come out of GWAS, and then you look to see where in different tissues those risk genes are concentrated on sites of epigenetic activation. It turns out there are only two tissue types for which that is true. There's the brain, which is not surprising, but quite reassuring. So in a lot of brain tissue, but particularly fetal brain tissue, epigenetic activation is co-located with risk genes for psychiatric disorder, and these are trans-diagnostic GWAS risks. So these are the risk variants that confer increased risk across a range of different psychiatric disorders. They're all co-located with epigenetic activation in fetal brain particularly. Pancreatic islets is the only other tissue besides the immune system. And in the immune system, it was particularly these T cells, parts of the adaptive immune system, where you had co-location of epigenetic activation and inherited risk. And Mary Ellen did the same analysis, but replicated it in a different epigenetic data set and looked not just at trans-diagnostic risk, but what we call cis-diagnostic risk for specific psychiatric disorders, schizophrenia, depression. She also looked at GWAS for BMI and for Alzheimer's, and you can see for trans-diagnostic risk, schizophrenia, depression, and BMI, in all cases, you've got some evidence of significant co-location of epigenetic activation with the relevant GWAS variants, whereas for Alzheimer's, it's a different story, and the risk variants are co-located with epigenetic activation in myeloid cells rather than lymphoid cells, the adaptive immune system. And those data and other data allow us to start thinking about this kind of model for what might be going on. So you've got some kind of environmental impact. It could be infection, it could be social stress, which induces epigenetic activation. And if the individual concerned has inherited risk variants for psychiatric disorder, activating those genes may change the phenotype, in the first instance, of T cells or other adaptive cells with long memory. Those T cells may control the activation of myeloid cells in the innate immune system, causing increase of cytokines, CRP, and so on, in the periphery and in the brain. And the blood-brain barrier is shown here, but it's far from an impermeable barrier. So a lot of things going on in the periphery, in particular cytokines and indeed immune cells, can traffic into the brain and modify neuronal function in that way. You might also imagine that there could be sort of effects of psychological stress, in particular refracted through risk variants and directly impacting on T-cell phenotypes in the brain. So this all needs unraveling experimentally and is quite complex, even if you think about it in a kind of snapshot way. But actually, you also have to think about how this evolves over neurodevelopmental time, because there's increasing evidence, which I'm not going to go into in any great detail, that immune cells are important in neurodevelopmental changes, particularly synaptic pruning. There's a lot of evidence that microglia, which are kind of phagocytic cells, can literally eat synaptic terminals and may contribute to synaptic pruning in that way. So there is a developmental angle to consider as well. And that gets me to the third bit, which is trying to think about the, what's going on in the brain during those first 2 decades of life. What are the kind of processes that an abnormal immune or an atypical immune state might be impacting on? And this is a paper from Richard Bethlehem and colleagues that was published earlier last year. This is about 120,000 MRI scans over the entire life course from before birth through to about 100 years old. This is a log scale for age, and these are the trajectories of development in terms of fairly simple brain imaging phenotypes: grey matter volume, white matter volume, subcortical volume, and so on. And the key point really is that the period between mid-pregnancy and early childhood is absolutely critical period for brain development. But for some phenotypes, particularly white matter and subcortical volume, there's ongoing developmental changes throughout adolescence and indeed into early adulthood. And that time frame is obviously coincident with and maybe plays a causal role in the emergence of so many mental health disorders over the same period of time. And I'm just going to show you some other data pointing to how genetic, sex, and environmental effects could impact on those developmental trajectories. This is a study from Eva Maria Stauffer, who's a PhD student in the lab at the moment, looking at the genetics of schizophrenia from GWAS data again, calculating polygenic scores for risk of schizophrenia, and then investigating how those polygenic risk scores are associated with imaging phenotypes, and finding some pretty robust evidence for increased polygenic risk of schizophrenia being associated with decrease in this microstructural MRI marker of neurite density, which is an index of connectivity at a sort of microstructural level in the brain. So risk for schizophrenia turning into abnormal connectivity at that level. But it's not just the genetics, and this actually is where I think it gets most exciting for the next generation. It's the intersection between what you might call psychosocial risks and biological risks for mental illness. So these are normative results looking at magnetization transfer, a measure of intracortical myelination in a healthy adolescent cohort. So at the age of 14, the bits of the brain that are most strongly myelinated are, of course, the primary visual, primary motor areas, and then over the course of adolescence, most of the additional myelination occurs in association cortical areas. But what's interesting is when you look at the rates of the within individual rates of change in cortical myelination during adolescence, stratified by socioeconomic status as a proxy for early life adversity, social deprivation, you can see there are differential rates of intracortical myelination in this frontostriatal system, depending on whether people were born and brought up in low or high SES backgrounds. That's kind of interesting to me. And I think there is a lot still to do in terms of understanding how social factors play into brain development. Even if you don't buy all the immune stuff, I still think that's a very important question. It's just worth noting in passing that if you look at those 120,000 scans that Richard and colleagues analysed for the brain chart paper, or you look at the cohort here, which we collected in Cambridge and London, and you look at their socio-economic profile, it is massively biased towards affluence. So this is a sample that's most normative neurodevelopmental data sets are impoverished for poverty, if I can put it that way, or enriched for affluence. We're not including the population on a representative basis. So I think we do need to get a little bit more epidemiological in how we think about large-scale neuroimaging studies, and we probably need to make more deliberate efforts to be inclusive of traditionally underrepresented groups, which include people with low SES status. Sex is also important, and this is work from Leonard Dorschmidt, another PhD student in the lab, and these are functional MRI data. It's quite a busy slide, I apologise for that, but basically she's looked to see where in the brain are there sex differences in the development of functional connectivity. And it turns out that there's a set of regions shown here where females have what we call a more disruptive profile of developmental change. In other words, these are all regions which have relatively low connectivity at baseline and become more strongly connected over the course of adolescence. So these are, you might think of them as new functional connections coming online in this cortico-subcortical system, particularly in females during adolescence. And Lena did a lot of work trying to contextualize that result, showing that this system, which is developing differently in females during adolescence, is specialized for reward and other psychological processes that might be relevant to depression. It is correlated or co-located quite strongly with areas of the brain that are different in MDD cases compared to controls. And she used the Allen Human Brain Atlas to find a gene expression profile that was correlated with this phenotype, and was able to show that there was such a gene expression profile and that it was enriched for risk genes for major depressive disorder. So there's nothing about immunology in this slide. This is just showing that when we think about how the immune story is going to play into brain development, we need to think about the genetics, we need to think about sex, we need to think about socioeconomic background, and it's going to be a rich and complicated story to unravel in totality. Now, I said at the beginning I wasn't sure whether this was going to come to a conclusive point, and ultimately that's for you to decide. But what I would say is I'm not alone. So, in fact, I'm not even the first person to start thinking about this. There is papers by Robin Nusslock and Greg Miller in particular that have been pursuing this idea of a neuroimmune network theory for psychiatric disorders for some time. And you can see here the concept is similar to what I've been, or some of the ideas that I've been pushing today. You've got an early life stress exposure, which has effects on the immune system as well as effects directly on the brain, and that carries over the course of development, modifying neurodevelopmental trajectories so that you end up with an adult state, slightly atypical brain network organization, and increased risk of behaviors and experiences that may either constitute a psychiatric disorder or put you at increased risk of psychiatric disorder. So the key thing here is thinking about the immune system as the conveyor of a memory of early life exposure, early life adversity exposure that modifies brain network development over time. So that's it, and these are the conclusions. This association between depression and inflammation, I would say, is beyond reasonable doubt. but questions remain about causality and specificity. Epigenetic activation, I think it's very theoretical. I mean, it's no doubt it's theoretically interesting, but I think there's increasing awareness that it might also be practically accessible as a mechanism for long memory of early life adversity. And when you think about how that plays out in terms of neurodevelopment, there are a lot of angles to consider that you can summarize in terms of this kind of neuroimmune network hypothesis. And I'll just finish by thanking the many people whose work I've summarized briefly today. I won't go through the names, but many of them are PhD students or postdocs in the lab, and I'm very grateful for their energy and brilliance in doing the work I've talked about today. So I'll stop there and happy to deal with any questions.
Are you happy to take your own questions?
Yes.
Okay. Any questions?
Oh, yeah. Yes.
Are there any neuropsychiatric conditions that are associated with a decreased immune activation?
That is interesting. So I think there's some evidence that schizophrenia might be associated with decreased innate immune activation in the brain. But that is, I'd caveat that by saying, what we really would like, in order to investigate all these ideas, what you'd really want is a readout of brain inflammation, not blood inflammation, and that is what we don't have that at the moment. You can see these inflammation-related changes in MRI markers, functional MRI connectivity, proton density, etc. But the only sort of specific marker of brain inflammation we have is through PET, and the available radio tracers for inflammatory neuroimaging with PET are very, they have a lot of problems. I mean, they're mostly not very specific, but it's that line of evidence. It's the PET imaging study of schizophrenia that people like Oliver Howes have been doing. If you wanted to check or search on Oliver Howes PET schizophrenia, I think that'll get you to a sort of literature that looks at brain inflammation, and there is some evidence there that it is actually reduced brain inflammation associated with schizophrenia. Professor Everett.
I think we articulated this compelling story. What I'm trying to get my head around is it the case that there is systemic peripheral inflammation and key molecules, immune molecules, or indeed immune cells get into the brain and change the network, and that's really the predominant mechanism? Or is it that there are people as well who can be depressed but without a systemic or peripheral manifestation, and that actually these changes in the immune and inflammatory mechanisms can happen directly in the brain itself, happen by glia?
Yeah.
Or is it both, or is it that if you're depressed, you're bound to have systemic changes in your body systems that will generate this immune or inflammatory signal?
Right. I would say it's the second of the, so I think there's evidence that you can have a systemic inflammatory state which propagates through the blood-brain barrier and causes either direct neuronal effects or secondary effects on the immune cells in the brain that in turn have neuronal effects. There's quite a lot of evidence, again, from animal models for that, where you can, obviously in an animal, you can deliberately perturb the peripheral immune system and then demonstrate that there's a propagation of that signal into the brain. So that is, that's certainly one, that's one mode. But to your second question, your second option is, could you have a sort of altered central immune status, which is not associated with peripheral abnormality of the immune system? that is, again, certainly true. That's certainly vindicated by animal studies. So you can, for example, rather crudely, inject cytokine into, the ventricular space in an animal. There's no peripheral perturbation, but you can see the same sickness behaviour changes result as if you'd injected LPS systemically. So the stimulus doesn't necessarily have to be either peripheral or central. It could be either in the animal literature. Where we are in humans is contingent on the point that I was just discussing, which is we don't really have good markers of brain inflammation. We have some. I've mentioned PET. You can also, of course, look at CSF. But these are still, we have very limited data. And in practice, it's very difficult getting either of those kinds of data from patients with mental health disorders. So we know much less about the central state. And then the third option was, could you have, was it systemic changes as a result of depressive or central changes? And I think that's probably also likely to be true. I mean, most of the, I think the closer people look at it, the more it seems that there's a very significant two-way interaction between the nervous system and the immune system. And I've highlighted the arrows pointing from immune to nervous system to mental state. But in principle, there's no reason why the arrows couldn't run the other way. You have mental state changes that induce changes in brain states and immune states. And again, animal literature shows that quite clearly. You can expose an animal to a psychological stressor and demonstrate that there is a sort of centrifugal effect on circulation of immune cells of certain types, for example, that follow a mental exposure. A psychological risk exposure. So I think the arrows can go both ways. Yeah. I'm sorry. Chap at the back and then you, please.
What might be the evolutionary interpretation of the state?
Well, so there are evolutionary stories about this, and they are they're like a lot of evolutionary stories. There's a little sort of just so element to them, So, but one of a theory is that a key survival risk in our history has been infection. Infection is related to trauma. Trauma is related to social conflict. And in every case, there could be a survival advantage in having a preemptive immune response. So the theory about how social stress increases or has evolved to activate the immune system is that, historically, if I'm involved in conflict or there's a risk of, I'm involved in a social situation which is at risk of escalating to conflict and trauma, there's an evolutionary advantage in having the immune system preemptively activated so that, the macrophages are all fired up and ready to deal with the infection when the conflict escalates into trauma. That's a theory. I don't know how plausible you find that, but that is, people have written at some length about that. I suppose it's just worth bearing in mind, social stress. What I mean by social stress here, particularly extreme social stress, loss of a parent in infancy, for example, or physical violence in the early years of life. That has in historical and prehistorical time, that is a major risk to survival, on par with biological stresses like infection. And, it's not, it's quite intuitive that there should be some immune response to try and to offset the survival risks associated with social as well as biological stresses in early life. But, as I said at the beginning, it is a little bit just so. It kind of sort of makes sense when you read it as a narrative, as a story. But, it's perhaps more challenging to test it really compellingly. I think if there wasn't any kind of genetic angle to this, that would discredit evolutionary theories quite a bit, in my opinion. So the ideas that are beginning to come up through this particularly this sort of epigenetic intersection between environmental stresses and inherited risks that modify the phenotypes of the immune system, that kind of idea. I think building that sort of evidence would encourage me to think more creatively about the evolutionary relevance of it all. Yeah.
Since inflammation is common in depression and neurodegenerative conditions, would treating depression help the symptoms of someone with a neurodegenerative condition or impact the pathology in any way?
Treating the depression?
Yeah, treating the depression alone.
In neurodegeneration?
Yes.
Well, if they are depressed, it can, of course, improve the symptom profile in that respect. But if your question is, would treating depression in neurodegeneration alter the immune state that might be contributing to the neurodegeneration, I think that basically the jury, I don't know that we've got a definitive outcome on that. I mean, some people claim, I'm not particularly convinced, but some people claim that conventional antidepressants, SSRIs, have anti-inflammatory effects, and that might be one of the ways in which they work. I'm personally not convinced of that, mainly because we did a little experiment ourselves to put that to the test, and it turned out that conventional antidepressants did reduce the inflammatory state of cell cultures, but only by killing the cells. So if you used a dose of antidepressant which wasn't cytotoxic, they didn't actually seem to modify the function of living cells. So treating depression certainly can improve the symptomatic state of somebody with a neurodegenerative disorder who is depressed, but I don't think it does it by an immune mechanism. Yeah. Mark?
Perhaps a related question. You mentioned that you weren't entirely confident about the evidence seeking to dissociate an improvement in mood because of the improvement of the underlying inflammatory disease.
Yeah.
Dissociate that from the idea that the anti-inflammatory would act directly.
Yeah.
But you mentioned studies in which groups of patients had been included or not.
Yeah.
And I wondered whether a covariance approach might be more powerful. We wouldn't normally do structural engagement modeling on a meta-analysis. You have several trials there.
Yeah.
But there might be a workaround for that problem. Well, did anyone try that?
Well, we did try various. We did try various workarounds. We did try some not exactly a covariance thing, but we did try and sort of statistically factor out physical health changes. And as I said, we also the workaround that I found most persuasive, perhaps because it's conceptually the simplest, was just to take a subset of patients who had not improved physically and show that they still had the mental health improvements. But I mean, really what it needs is longitudinal sampling of mood changes following the initiation of anti-inflammatory treatment in patients with rheumatoid arthritis and so on, because there is, when anti-TNF antibodies first came into practice, which is about 20 years ago, and that immediately changed the outlook for people with rheumatoid arthritis, a lot of practitioners noticed that something that they called the Remicade high. Remicade was the brand name for the first anti-TNF, or one of them, which is that, when you infuse an anti-TNF into somebody with rheumatoid, they very often experience a boost in mood. They become slightly euphoric within 24 hours or so. In fact, my old boss at GSK, who's Patrick Vallance, you might have heard of him, when I was talking to him about this, he said that when he'd been a medical registrar or consultant at UCH, the nurses had competed with each other to put the infusion into patients with rheumatoid because they knew that the patient was going to feel better and would thank the nurse, So it's kind of like embedded in the sort of clinical folklore that anti-TNF and other anti-inflammatory drugs can have very short-term benefits on mood that you see that are visible, obviously visible, long before there are any kind of X-ray changes or changes in, physical symptoms. But, all of the clinical trials all of those clinical trials had not measured any clinical endpoint until 3 months after the start of treatment. And, what you really need is to show, in the first 24 hours, in the first week, what's the mental state, what's the physical state, and the prediction would be that you see a mental state improvement that precedes and therefore can't be explained by the physical improvement. Yeah. Oh. Yes, sir.
Is there any evidence that an active coping response produces neuroinflammation?
An active coping response? Not that I'm aware of. But there is a literature that probably could be better, could be stronger, which is looking at how psychological state changes can have immune consequences. So people have looked at, for example, meditation or mindfulness training, and asked the question: if you deliberately try and change your state of mind, does that have consequences in terms of the state of your immune system? And it's a bit of a patchy literature, small n, usual story. I wouldn't say there's a convincing consensus, but it's an area where bigger, better studies could be quite interesting. Yep.
Given the general picture of kind of enhanced immune function and autoimmunity in women,
Mm-hmm.
Is the subgroup of MDD patients with immune overactivation more pronounced in women, or is there no sex difference?
Well, it does. I think there probably is a bit of evidence for that. And again, it might be the sort of Brenda Pennings is a good sort of search term if you're interested in this, because it's a rather unusual surname, and she has published quite a lot of epidemiological data on this, and I think out of her work you get the sense that there is a sort of clinical subgroup which is sort of middle-aged female, overweight, at risk of metabolic disorder, and also with these atypical depressive symptoms, and that I think that does exist as a sort of a concept, but I don't know. It's in our work. Certainly, when you look at some of these gene expression changes, they are very sex dependent. I mean, I didn't really go into that. The stuff I showed was kind of averaging data over different sexes, but if you split it out by sex, there are obviously big sex differences in gene expression per se, and then there are sex differences in gene expression related to depression. Yeah. So I think sex is going to be an important, moderating variable of all of this.
Now, I know that, Ed, you need to go to get a train to London, so I think we probably need to finish the questions there. But can I just ask one more question?
Yes.
In relation to that? I wondered, your question, really interesting about sex differences, I wondered how hormones interact with immune system. Whether there's anything known about that and results.
Do you mean sex hormones?
Yeah, like estrogen in particular.
I don't know much about that, no. I mean, obviously you've got the glucocorticoid, the whole HPA axis, and the anti-inflammatory effects of glucocorticoid receptor activation are very there's a very tight tie-in there between neuroendocrine and peripheral immune states. But I don't know so much about
I asked
the sort
the major sex differences in for example, depression.
Yeah.
Don't worry,
Yeah.
it's just we'll deal with that. In sex differences in depression and other internalizing disorders occur at puberty.
Yeah.
And one theory is that there are various factors associated with puberty, both social but also, of course, hormonal. And I wondered whether, for example, in your study where you're talking about sex differences and brain development and immune markers, whether hormones have been measured.
Sadly not.
Okay.
No. But it is an interesting angle. Yeah. And I wouldn't be surprised if there were sex hormonal interactions as well.
Okay. Thank you for a fascinating lecture.
Okay, thank you.
Hormone scanning drug directly with the immune system. They have receptors, many different secretions, and they can interact. Plenty of papers on it, especially during pregnancy or supraphysiological hormonal level of direct influence.
Very interesting.
Well, there's plenty of papers.